This is a response to NephilimFree's video overlapping and embedded genes. Hello again, Neph. It's been a while since I addressed any of your malodorous videos because I felt it only fair to let you recover from the double fisting I gave you in Holy Hallucinations 9 and 10. But now that you're hopefully feeling a little less dilated, I thought it was high time that I paid you a friendly visit and put you straight on one or two little inaccuracies I found in your video regarding the nature of overlapping genes. Now, anyone who's had the misfortune and intestinal fortitude to watch one of your originals from start to finish will already know of your prodigious talent for taking 10 minutes to haphazardly stumble through what can be said in 2. As a result, there may be some repetition in the clips I'm going to show from your video, but that seemed to be the only way in which I could present the occasional new fecal nugget that you intermittently introduced during your seemingly endless regurgitation of the same point. But before I play the first one, you should note that if you happen to think that you're sounding a little more eloquent than usual, it's because I took the time to edit out the ubiquitous pregnant pauses that intersperse your craptodidactic prose like peanuts in a squirrel turd. It's hard to watch your videos without repeatedly banging your head against the keyboard until your ears bleed, Neph, and so I decided to do my best to shield my subscribers from sharing the pain. Take my advice. If you're doing it because you're laboring under the impression that it makes you sound thoughtful or intelligent, then you're wrong, because the effect's eerily reminiscent of an intellectually impaired lobotomized chimpanzee with a concussion. And if you're not doing it deliberately, then might I suggest a script, an audio processor, and just a small modicum of pride in your work. Radical thoughts, I know, Neph, but they really will help alleviate the impression that you're moonlighting as a stand-in for Forrest Gump. So now that I've finished impugning your intelligence, let's get on to the part where I factually demonstrate that these weren't ad hominem attacks, but rather a reasonably accurate description of observable reality. You may want to go and find something to bite down on while I play the first clip, Neph, because this is going to sting a little.
I explained to her that one of the problems with the idea that random genetic mutation has developed in the genome of all the life forms of the Earth, and is the stuff of life for evolution, is the fact that we have discovered that genes are often overlapping and embedded.
Okay, Neph, now that we have the basis for your thesis, I'll take a little time to describe the general structure of eukaryotic genes. This will make it somewhat easier for anyone watching this video to follow what I'm saying when I get to pointing out exactly and precisely why you're full of crap. In general, bacterial genes are stretches of DNA containing an uninterrupted linear sequence of coding information for their respective proteins. By contrast, most mammalian genes aren't contiguous and their coding regions are broken up by islands of non-coding sequence known as introns. After transcription into RNA, the exons containing the coding information need to be joined by removal of introns in a process named splicing that occurs in structures named spliceosomes and can be envisaged as being akin to excising adverts from a movie before watching it. This mature messenger RNA of fused exons can then be translated into the final protein by ribosomes. Since the genetic code is read in triplet form, where a codon of 3 consecutive bases represents 1 amino acid in the encoded protein, each mRNA can be read in 3 potential reading frames depending on where translation is actually initiated. The actual region of translation is delineated by stop and start codons. Since these are seldom located at the ends of the transcript, not all of the original exonic material codes for protein, and the non-coding ends are named the 5' and 3' untranslated regions. So now that I've laid the groundwork, let's get on and take a look at what kind of monumental lies a creationist can conjure out of thin air by just reading an abstract or two, praying to his ephemeral special friend for inspiration, and willfully ignoring anything that even vaguely resembles a fact. But before we do that, Neph, I was curious by what you meant by the phrase "we have discovered" when referring to overlapping genes. In fact, you seem to be very fond of using this particular conjunction in this video, and if you don't believe me, then take a look at this.
We will continue to discover. We have discovered. So far we have discovered. We may discover. We continue to discover. We've discovered. We continue to discover.
Of course, the reality, Neph, is that you couldn't discover your own arse in the dark with both hands, let alone anything in the field of molecular genetics. I found this particularly galling because the people who actually did the discovering that you're referring to are the same people who'd piss their pants with laughter if they ever heard you ignorantly opining on the subject that they've spent decades working in. I've said it before and I'll say it again, your 2 years of typing evolution is a lie into Google and voraciously lapping up anything it spat back at you like a pig at a trough full of swill doesn't make you an expert in anything other than dishonesty and gullibility. The quality of your online education's evident every time you open your mouth, and no more so than in the next clip.
This illustration demonstrates why all genetic mutations cause degradatory effects to the genome's code. Because if a random genetic mutation which affects this portion of the gene takes place, it will affect this gene. So even if we were to find that a genetic mutation provided some imagined benefit to this gene, which is exceedingly rare, it is almost certain that it would cause degradatory effects to this one. Because genes are overlapping and embedded, random genetic mutations typically affect more than one gene at a time.
I was puzzled when I first saw this, Neph, because the figure you're referring to isn't annotated in English. It turns out there's a good reason for that, and that's because you found it on a Finnish creationist website. It seems that you're broadening your horizons, Neph, which is nice, since all other indications are that you really don't get out much. Now, this diagram of yours bears no semblance to any genetic locus I've been able to find, and since your Eurotrash website, of course, didn't give any indication of a primary source for the figure, I think it's safe to assume that it's been concocted for the sole purpose of allowing shamelessly dishonest reptiles like you to misrepresent the complexity of overlapping genes. Your little artistic interpretation, though, does illustrate the basic concept, and that is of a genetic locus containing 2 or more genes that share at least some of the same physical sequence. An overlapping gene can reside on the same strand of DNA as its partner and be in the same or different reading frame, or more commonly on the opposite antisense strand. Because of the anti-parallel structure of the DNA helix, same-strand overlaps always run in the same direction, while antisense overlaps run head-to-head or tail-to-tail. Additionally, one gene can be embedded entirely within another, and when it resides within only one of its partner's introns, it's referred to as nested. Now, despite your mendacious assertions, Neph, there are many reasons why the restrictions on the mutation of overlapping genes aren't quite as stringent as you would have the poor people you're trying to deceive with your vile lies believe. And now I'm going to proceed to ram those reasons up your jacksie one by one. 1. Just because genes overlap doesn't mean that they share exons. The coding sequences of nested or embedded genes are entirely contained within one or multiple introns of their hosts, and Makalovska and colleagues showed that these represent over 50% of overlapping human genes. Since mutations in the embedded gene only affect the introns of its host, they have no effect on its coding sequence and so aren't subject to the restrictions you're talking about. 2. The genetic code is redundant. The 3-nucleotide 4-base system produces 64 possible codons so that all of the 20 amino acids are represented by more than one. As a result, any given point mutation may be synonymous and result in no change in the protein sequence in one gene of an overlapping pair, but may concurrently be a missense mutation that alters the peptide sequence of the other. 3. Not all missense mutations result in an alteration in protein structure or activity. Such conservative mutations substitute one amino acid with a structurally similar one which, depending on its structural context in the protein, may not produce any functional change. This kind of mutation in one gene may not be so innocuous in its overlapping sibling, allowing the 2 genes to diverge. 4. 75% of antisense genes don't code for protein products and may function to regulate the transcription of their cognate partners. In this instance, mutation in either gene would by definition not be restricted by the other. 5. Gene duplication facilitates the generation of new copies of the overlapping genes and the subsequent unrestricted generation of mutant alleles in the presence of a functional second copy. I'm not surprised that you neglected to mention this little tidbit, Neph, because by now creationists are well versed in deliberately ignoring this well-documented and understood process whenever they spuriously claim that mutations cannot generate new information. And 6, in the absence of any of these mechanisms, you'd still be wrong because there's a problem with your argument that's so big that even someone as astonishingly simple-minded as Shock of God would be able to see it. But I'm not going to spill the beans quite yet, Neph, so let's just let that thought sink in and let the tension build a little. Meanwhile, I think I'll finish up on this point, Neph, since you might be feeling rather full. So while you're recovering from the rectal overload, let's watch you frenziedly flogging your dead horse again as if your friend's disability check depended on it.
But the point is that random genetic mutations are degradatory to the code of the DNA molecule, and overlapping and embedded genes is one of the reasons they are so degradatory. because a random genetic mutation which occurs to one gene very well may affect another, or another and another, or another and another, and another.
Okay, Neph, I'm not going to bother responding to your deliberately erroneous assertion that random mutations are degradatory to the genetic code, because that's already been roughly stuffed back up your arse more times than your router's been connected to the erosguide.com server. Instead, I want to focus on the number of times you used the word another. I counted 6, so with the first gene you mentioned, that makes 7 sharing at least one codon. I've yet to read a single report of even a triple gene overlap, so I think it's fair to say that you are being a little less than accurate, presumably in a transparent attempt to again exaggerate the limitations of gene overlap on the generation of diversity. The only thing I'm having a hard time with is distinguishing whether you said this because you are still basking in the afterglow of a particularly vicious cerebral infarction, or because you're just a dishonest little piss stain who's willing to say or do anything to maintain his delusion that he actually has some sort of argument to make.
Sophia, you're behind. Genes are indeed overlapping and embedded. As time goes on, we will continue to discover more and more and more of these overlapping and embedded genes. So far we've discovered hundreds. Before we're through, we may discover many thousands.
As usual, Neph, your assertion is nothing more than a pathetic and easily exposed lie, though I can't be sure whether you said it deliberately or inadvertently because your head's permanently wedged up inside your colorectal cavity. So let's examine your claim that we're only just scratching the surface of the embedded gene count by taking a little drive down memory lane. Prior to the invention of DNA sequencing, it was thought that one gene coded for just one protein. But following the sequencing of the first DNA genome, bacteriophage 5X174, by Fred Sanger's group, a feat which played a central role in him winning his second Nobel Prize, it became clear that there were exceptions to this rule. Subsequently, overlapping genes were discovered in many viruses and are elegantly explained as an evolutionary strategy to minimize genome size due to the size restrictions imposed by capsid geometry. Later, overlaps are also found to be relatively common in bacteria, which are also under some evolutionary pressure to restrict genome size. As an aside, isn't it interesting, Neph, that these observations are perfectly understandable in the light of evolutionary theory, whereas your alternative, God did it so he could be dark and mysterious, leaves you as ignorant as a sad bastard with a pretend degree. Now, getting back on track, it was therefore surprising that during the late 80s and 90s, handfuls of overlaps started turning up in eukaryotes, which are thought to be relatively unaffected by such selective pressures. Then, following the sequencing of the human and other genomes, it became possible to search for overlaps using algorithms that predict the presence of open reading frames. That's where those hundreds you mentioned came from back in 2004, and there's been no significant change in the numbers since because there are no more genes to be found. Your projection, therefore, of there being thousands more to come is about as ambitious as Bill Dembski's and William Myers' hopes for a Nobel Prize, because after all, it's now 2001 and we're still waiting. So with that behind us, we now get to the point I've been looking forward to for the whole of this video, Neph. So tie down your headphones, grit your teeth, and hold on tight, because this one's going to make your eyes water.
According to this paper, thus far are discovered 774 genes overlapping in the human genome, and 542 genes overlapping exons in the human genome, compared to 578 genes which are overlapping in the mouse genome, and 455 genes with overlapping exons in the mouse genome. Notice the difference in the number of overlapping genes. The discovery of overlapping genes is just in its infancy. As time goes on, we continue to discover more and more genes which are overlapping, or embedded, or both.
Of course, this last statement only serves to demonstrate that the only thing that's in its infancy is your intellectual development, Neph, because I just explained why your claim about the future trends of overlapping gene discovery is about as accurate as a Kent Hovind tax return. The mistake you made here was using an actual paper instead of just showing us your usual series of screenshots of abstracts, layman's science pages, and creationist websites. So the first thing I did was track down the actual document and do what you obviously never did, and that's read it. And having done that, I'm now in the happy position of being able to use it to beat 7 shades of shit out of you. You see, Neph, while your numbers might sound impressive to someone who didn't know any better, they did leave out a minor point. And that's that these genes were found in genomic databases containing 34,604 human and 33,936 murine genes. Now I'm assuming that long division isn't your strong point, Neph, so I'll do the math for you. That means that of all the genes examined, only 2.2% were overlapping in humans and 1.7% in mice. Hardly the impressive figures you are making them out to be, eh? But wait, it gets better. Genes that don't contain exonic overlaps don't count because mutations and overlapping introns have no effect on protein sequence and therefore aren't subject to the mutational restrictions you're claiming are a death knell to evolutionary theory. So when you only count the genes that contain exon overlaps, you come down to values of 1.6% in humans and 1.3% in mice. Not looking any better for you, is it, Neph? And if you think that's bad, then all you have to do is read the rest of the table you showed us and think back to something I said about exons earlier in this video. Remember that I said that parts of the exon sequences in the mature mRNA weren't actually translated into protein and therefore weren't coding. Well, it turns out, Neph, that the vast majority of even these exon overlaps are in non-coding regions. In the end of the day, only 57 human genes overlapped in coding regions and 31 in mice. That works out to be less than 0.1 to 0.2% of all the genes in the human or murine genome, Neph, or as we trained scientists like to put it, absolutely fuck all. I'd love to know how you managed to miss this, Neph, I really would. Was it because those difficult long words in the paper were just a little too much for you? Or because you chose to ignore it because the truth was just a little too inconvenient? Well, now that I think I've unequivocally stuffed your ideas back where they belong, I should start wrapping up, as this has already turned out to be a lot longer than I'd hoped. And while there are a number of other annoying putrid flecks of misinformation sprinkled throughout your video, there was one that I felt I just couldn't let slide. So let's take a look at it, and then I'll let you get off to the pharmacy for a little relief.
Geneticists today are shying away from the use of the word gene because it really doesn't properly describe what we observe in the genome of life. The preferred term is sequences.
Do you really believe the mind-bending inanity of the verbal spew you bring up every time you open your mouth, Neph? That's a rhetorical question, of course, because I think it's quite clear from your videos that you've a misplaced ego larger than Joel Osteen's bank account. Of course, this is complete and utter bollocks because the word sequence means what it's always meant to trained and qualified biological scientists. That is, the conceptual representation of the linear order of bases in any given segment of nucleic acid, while the word gene refers to a unit of heredity that is physically manifested as a specific region of DNA with a specific sequence. Despite your demented rambling, Neph, nothing's changed. Just because an ignorant and intellectually compromised bozo asserts the contrary with the expectation that his hallucination will be unquestioningly accepted doesn't change the definition of these words. It only demonstrates his profound arrogance and the magnitude of the gulf that separates his perception from that thing that the rest of us refer to as reality.